DNA isolation procedure from blood broken down step by step using dots for clarity. Take 200 to 500 microliters of whole blood in a microcentrifuge tube. Add an equal volume of PBS and mix gently. Centrifuge at 5,000 revolutions per minute for 5 minutes to pellet the cells. Discard the supernatant. Add 500 microliters of lysis buffer containing SDS and proteinase K. Mix gently and incubate at 56 degrees Celsius for 1 to 2 hours, or overnight, to digest proteins. Add an equal volume of phenol chloroform isoamyl alcohol, 24 to 1. Mix by gently inverting the tube. Centrifuge at 10,000 revolutions per minute for 10 minutes. Carefully transfer the upper aqueous phase to a new tube. Add 2 volumes of cold 100% ethanol to precipitate the DNA. Mix gently by inversion until DNA forms a white precipitate. Centrifuge at 10,000 revolutions per minute for 5 minutes. Discard the ethanol supernatant. Wash the DNA pellet with 70% ethanol. Centrifuge again and discard the ethanol. Air dry the DNA pellet briefly, do not overdry. Resuspend the DNA in 50 to 100 microliters of Tay buffer or sterile water. Allow the DNA to dissolve completely, ready for downstream use.